All right, everyone. Thank you guys so much for coming. We're going to get started. I'm really excited about this event. Just get you guys on the same page. I'd like to read our mission statement real quick, just to give you an idea of what this is all about. So the Discovery Cafe supports the betterment of society by using trust-building events to facilitate mutually engaging connections between scientists, researchers, and Western New Yorkers. And I also want to take the time right now to thank SUNY Geneseo, especially the Center for Inquiry, Discovery, and Development, for not only helping me to start this event, but they're also continuing to support it in the spring semester and next year. So can't thank them enough. And the reason why I'm really excited about this event today is because I'm introducing Dr. Travis Bailey from the SUNY Geneseo Department of Biology. Not only is Dr. Bailey my professor, he is also my academic advisor, my research advisor, and just a mentor on campus who has supported me immensely even with this project. So I can't thank him enough, and I'm really honored to introduce him. So without further ado.
Patrick made it rather obvious how I bragged my way into this. But I do want to thank Patrick and David Parfit, who's around here somewhere, and his Center for Outreach that's made this evening possible. I'd like to start a little bit with talking about who I am, so you know what the Joker is at the front of the room talking to you is about. I'm from Utah. I got my undergraduate degree at Brigham Young University in Provo, Utah. From there, I went to Baylor College of Medicine, which is a medical school down in Houston, Texas, and a lot of people confuse it with the Bears. It's not the one in Waco. It's a different school. Both of them are good, though. So, but I felt like if I would have gone there, I could have gone to the Baptist school too, because from there I went to Notre Dame, went to the Catholic, and this is the first time I've been in a secular institution. I'm married. I have 4 kids. 2 of them are here. One's in the front row. That's Megan. Zoe's 3rd row back. And they brought a bunch of their friends from a congregation here that meets over on South Street. My wife couldn't be here tonight because she's picking up my mother-in-law from the airport with my 2 youngest daughters. And so just the way things work out sometimes. But I moved here about 4 years ago to accept a job in the Department of Biology, and love it here. I love the people that I work with, mostly because the department, I know, even if we disagree about things, it's always about what's best for the student, not what's best for the professor. And I haven't got that with other places that I've been. So I really like being here. I like the gorgeousness of Western New York and tromping through waterfalls and everything that this area has to offer. I'm also kind of a casual type of person. Sometimes I don't seem as academic because of the kind of ways I use to express things. But please feel free to interrupt me at any point during what I'm talking about if you think that I didn't say it loud enough. My voice goes about 5 feet and then drops off. Usually I'm mic'd in the classroom. But if you're confused about what I'm saying, you want a clarification, you don't need to wait till the end and remember your question then. Just stop me anywhere in the middle of it. On the table up here, I'm going to show you something that I work on. These are what are known as zebra fish, or Daniel Ririo. You can find them in Walmart. If you want to buy some, they're just not the kind that I use. They're just a little bit different. So I invite you to come up afterwards to see what they look like. They're about 1 inch long when they're adults, but I brought some babies, some of which were born just today. And you'll not be able to see them very well because they're dinky. I mean, they're really tiny. And I didn't have time to bring over a microscope for you to look at them. I won't speak a lot about my particular research today. I've only got 2 slides on it, because what I want to do today is to talk a bit more about stem cells in general, and not just the part that I work on, but the whole field as it exists right now, and to give you a bit of an understanding about how we think about the way life develops and the way things go wrong, and what kinds of opportunities we have to see if we can alleviate suffering, and what are the challenges that we still have, what problems still exist on making that wish a reality. So my title is Stem Cells, Challenges and Promises for Medical Therapy. I'm going to try and touch on a bunch of them in turn. I should also say I'm a member of the SUNY Eye Institute, which is a consortium in the 47 SUNY schools to do research on how eyes develop and how they regenerate. I'm pretty much the regeneration side of the ring, but I work on development as well. All right, as a stem cell biologist, we need to give you an idea of what we think about. One of the things that we're going to talk about today is why we need stem cells. And what I mean by that is not why we're going to need to develop them, but rather why we already have them in our bodies and how we use them just after we've eaten lunch to survive this world. I'm going to tell you about some different types of stem cells, ones that you probably already think you know about, and hopefully some that you don't really understand yet. And I'm going to explain them by telling you what their abilities are, how they function, where they show up. And in doing that, what I'm going to tell you about is what they can't do, and why we have no good panacea for the things that we'd love to just say, okay, you're sick, let's give you some stem cells and make you well. Okay, so if I don't teach you anything else tonight, what I want you to understand is that there are 3 different types of stem cells. And I'll take some time to go through all of them. But we can tell there's 3 different types because we find them at different places in our bodies, and we find them at different times of our life. So if we can distinguish time and space about these cells, then you'll be able to understand what I mean by that. The problem with them is they're more or less flexible in being able to do things that we want them to do. And by more or less flexible, I usually mean less. And what we're trying to do is get them to be more. And if we can make them more, then there's some possibilities that we'll be able to use them for. We need stem cells every day of our life just to live. You are not the same person that you were 7 years ago. There's very few of you that have the same cells that you were 7 years ago. And some of you, that might be the reason why you don't remember 7 years ago. But for the rest of us, some parts of us will turn over even faster than that. So we need stem cells to be making sure that we're always replacing cells that we're losing at all times. So what that means is that we're going to maintain some continuity of our tissues. In addition to that general just replacement of cells that are naturally going to be lost, we sometimes do crazy things. We fall off the roof when we're skating, or, no, I haven't done that. But I have fallen off my bike and ripped my face off. And that was decently traumatic, and I needed to somehow put all of the skin back on my face so that you don't get nauseated just looking at me. I'm sorry you're already doing that to some extent, but I'm really being nauseated. So you probably have already understood this, but some of your tissues do a pretty good job of renewal. They repair, they regenerate, all right. But there's other kinds of tissues that you just don't get to regenerate very well. And I'm going to talk a little bit about what we're trying to do to help some of those lagging tissues become a little bit more robust. In general, the plan is, can we farm some of these cells? Can we go over and treat a few of them right and make them multiply into large numbers and then get them to act like we want to? And then maybe we can put them back into somebody and get something to work out. In the jargon that I'm going to use today, we call that culturing the cells. But really, we're just trying to industrialize the process. All right, so I gave you the one that you probably already know about. Your skin's pretty good at regeneration. Every day, you lose about 1 gram of your body in skin. Not because you did anything wrong, but just because you woke up and you moved around and then you went to sleep that night. And so you have to replace those cells every day. And this is what it looks like if you took your arm and you cut it in a cross section and then you just look at the most superficial layer. So we're not getting down to the bones, we're not getting to the muscles, we're just way up in the skin. Skin's actually divided into 2 layers. One we call the dermis, which means skin, and the other one we call the epidermis, which means the above skin, which means skin. But anatomy lessons aside, these cells I'm not going to pay attention to. They're not the ones that I'm going to worry about, but the similar kind of process goes on with them. What I want you to pay attention to is these cells above that are just drawn in cartoon form. There's two layers, the epidermis and the dermis, and there's an interface between them. And at that interface, there's just this one layer of single cells. And those single cells are stem cells. Now we don't know which one is a stem cell. They all look alike, but some of them divide, and the other ones kind of hang out. And the ones that divide rather slowly. Some of them take about a year to get their act together and make a new one. But once they divide, the daughter cells multiply very rapidly. So if we're not looking carefully, the stem cell we confuse with the multiplying cell. And the multiplying cell is not a stem cell. I'll explain why it's not a stem cell in a couple more slides. But for now, just remember, it's the one that's faking you out. It's not the real stem cell. This daughter cell will move up into this area here, and then divide again 1 or 2 more times. But after she's divided 1 or 2 more times, she stops dividing. But the stem cell divides again, and a new progenitor cell that's multiplying rapidly will divide behind it and shove her up. So if you look at this picture, what I'm trying to get you the idea is that the first cell is here, but it gets moved and then gets crowded out by a new group of cells behind it. And so it gets pushed farther and farther to the surface of the skin. And over time, it changes what genes it's turning on, and it changes its overall function until it becomes pretty much dead. So yes, everybody that you've looked at, the most attractive person you have in your life, when you look at them, you're looking at the most dead part of them. You're looking at their skin.
You?
Yes. Well, it's also the reason why if your vacuum cleaner gets a little overheated or your heating elements get a little overheated, that it stinks in your house. It's because some of those skin has made it on the filament and started burning. And we don't like the smell of burning flesh, right? It's not cool.
Mm-hmm.
Those of you that actually touched your finger to the oven know what I mean by that. So every day, 1 gram of these cells are falling off, but we're replacing them with a new gram of cells that come behind them. And because of that, we don't develop any holes, we don't start bleeding all over every place, and we're able to keep bacteria on the outside, and we can go swimming and all sorts of things without any problem. This is normal. This is the way things are, and the way we wish all of our organs could be. Because if all of our organs acted like this, we'd be pretty good at, almost living forever. Because this just keeps going on. Even rather old people have pretty intact skin. And that's not their problem, what finally causes them to leave this world.
Travis,
Mm-hmm.
what's the timeline for that?
Let's see, if I remember right. It's going to take about 6 months. So your skin turns over decently rapidly. Cells in your gut turn over about every couple weeks. And this is why when you have chemotherapy, you have problems with your intestines. It's because you're killing off a lot of the cells that you're trying to replace them. Now, these aren't cancer cells. They're proliferative, though, and your cancer drugs oftentimes hit those. Okay, so let's just talk about the skin a little bit more. The skin cells seem to know that they shouldn't be dividing anymore, the stem cells, when they come in contact with each other. And we can change that. What we can do is see that these cells have nuclei, lots of little dots here, and then we can also see that they've got edges in the black. And I'm sorry this is just a cartoon about it, but if you can look at that, you can see that they're not really proliferating and they're just kind of sitting there. And also I'm going to see whether or not my movie actually started. Nope. Let's try it again.
Fibroblasts grown in vitro in a culture dish form a confluent monolayer of cells.
I want to
Cells
talk
in
to
a
you about
monolayer
that.
are relatively static. Contacting each other inhibits their migration. Such cell layers
okay.
can be wounded experimentally.
I'm going to have to talk
by
over
scratching
her
them
head,
with a needle.
but now I'm going to take a knife and cut the cells. If I do cut the cells, then what happens is that
In such an
it says there's a
we
wound
can observe
and it
that the
needs to
fibroblasts at the edge of the
engage.
wound become migrative
So the stem cells,
and
which
quickly
were
move to repair the
inactive,
gap.
all of a sudden become quite
Such
active.
cell migration is important for
It
wound
doesn't let
repair in
me
an
actually
intact
turn the
organism.
sound off. Sorry. I have to have some other source to do it. Let's see if I can do it this way. Over time, though, those cells that were just sitting around on their butts, they get off their butts, they divide, and then they grow together again, and then they slow down again. So somehow cells have the ability to tell when they're needed and when they're not. This is healing. This is regeneration. And it's a really important process that we have in our bodies. I study another kind of adult stem cell. We call them adult stem cells because they persist in your adult form. They're cells that will regenerate your skin all throughout your life. But I study some stem cells that exist in the eye. I'm interested in neurology and how brains work and how they mess up. And so when I went to graduate school, I got myself involved in how eyes develop and what kinds of genes need to be turned on to tell a cell that it needs to be a photoreceptor cell or one of the other kinds of neurons that exist in the eye. So the eyeball is not a whole set of neurons. The neurons only exist at the back of the eyeball in a place called the retina. So when you're seeing something, then light has to come through this skin that's on the outside of the eyeball, which is called a cornea. And we actually have corneal transplants when people have some kind of opacity. That is to say, that it clouds up. But it's a nice thing that we can replace decently easily. Then it becomes clear. The light can be focused with the lens on the back of the retina, and people can see again. Unless they're people that suffer from neurodegeneration. Something like retinitis pigmentosa or glaucoma means that the cells that are at the back of the retina, they die off. And then it doesn't matter how clear your lens is or your cornea, you will never see again. So my research has to do with the organization of the retina. And what I'd like to do is zoom in on that tissue so that you can see the cells that I'm talking about. When we come down to the microscopic level and look at the eye, what I'm hoping you see is a bunch of blue cells. The ones that are at the bottom are the photoreceptor cells. They exist as rods and cones and can see colors if you're not colorblind. They have to talk to other neurons, which are these blue cells here, which then have to talk to other neurons, which are these blue cells here, which then have to talk to other cells in your brain so that you can actually form an image and perceive that you've seen something. So it's decently complicated, but you have to relay signals from where you see the light out to the brain. I'm not as interested in those cells as I am in this yellow cell that exists in the middle. This is called a glial cell. In the retina, it's called a Mueller glial cell. But for the longest time, people thought it was the garbage man of the retina. It would help clean up the messes of the neurons, and it might provide them with some of the food they needed. But for the most part, the blood that was coming through the capillary there should have done that job. So they're just the garbage man. What we found in our lab and some competitors' labs is that this cell here in the middle is actually an adult stem cell, but only for fish. All right, so these little fish that I work on, I chose to work on them not just because they're cute, but because they have this really cool ability to regenerate all sorts of tissue that we don't. You can cut their fins off, they'll grow their fins back. You can cut their heart into pieces, they'll grow their heart back. You can cut their spinal cord, and they'll regenerate their spinal cord. For a while, they'll sit at the bottom of the tank, they'll flop around, not doing much, but then when they regenerate, they'll get up and swim away. Christopher Reeve would have loved this.
Yeah, right.
Just one time off the horse, and he didn't ever walk again. But fish, you can do all sorts of damage to them, and they will regenerate. Most importantly, if you damage their photoreceptors so they become blind, they will engage this stem cell, and they will regenerate.
Does it only work with those fish or any kind of fish?
Not any kind of fish. I tried it with Medacula, and they don't work so well. So you got to get the right kind of fish. And I haven't surveyed a lot of them, but I don't know how many it can do. Now, we're not unlike zebrafish, though. We have those same kind of photoreceptors in the same area. We have the same intermediate neurons in the same organization. We have the same ganglion cells. We have the same connections to the brain. So at a cellular level, there's not much difference between us and fish other than they can also see UV light, where we're limited to just red, green, and blue. However, they're similar to us also in the fact that we can damage them. Your mothers, when you were small, told you don't go look and stare at the sun. In fact, during the eclipse, some decent physicists and other astronomers were telling you don't look at the sun, because if you do, you will go blind. This is because you can kill your photoreceptors. And if you kill your photoreceptors, you don't get to see again. I don't care what your glasses do. They're done being able to see. So you only get one shot at that. But the fish don't. We can take them and we put them in a room where we shine a really bright light on them. And poor things, they don't have eyelids and they don't have fins that can get in front of their eyes. So they're forced to look at the sun as long as we want them to. And we'll kill their photoreceptors off. And then what we can do is just look at them over time as they regenerate those lost cells. So what my research really is about is trying to figure out, how can we be more like zebrafish? Is there a way that we can get our Mueller glia to actually do this kind of thing? So just to show you what this looks like, we made a fish that will cause all of their Mueller glia to glow green. It's not a natural thing to do, but we can force them with some genetic tricks to do this. And the cool thing about it is it doesn't hurt them. They can live just fine with this new gene. So what we've done here is we've labeled all the Mueller glia green, and none of the other cells. So when we look at them, we don't notice any of the other cells. We only have to look at the Mueller glia, and everybody else just steps in the background and we don't see them. And if we look at the fish at 0 hours, meaning that we never turn the light on, so we never did them any damage, we won't see any of those Mueller glia doing anything but sitting there. But if we wait a little while and we look for those that are multiplying, we'll see a new marker come on in their nuclei. The one that's red. It looks yellow here because it's mixed with the green and red light look yellow to you. But over time, they turn off the genes that made them Mueller glial cells, and they just become multiplying cells. What's also cool about them is they still migrate. So they don't stay where the Mueller glia are, they move to where the photoreceptors were. And then they turn into photoreceptors. Let me show you that in cartoon form. So here's a normal fish. It's got photoreceptors, it's got Mueller glia. We shine bright light on them and we kill some of the photoreceptors off. The photoreceptors then send a signal to the Mueller glia and say, hey, we're dying, can you fix this? And then they die. And so they're not there anymore. But the Mueller glia start to multiply. And their baby cells make more baby cells. And then those baby cells migrate to where the cells were missing, and they differentiate back into the cells. Now this is cool enough, right? We can take a blind fish and it'll become sighted again in a little while. But what's extremely cool about this to me is you can take other drugs and kill off the neurons that are down here, and instead of trying to fix photoreceptors, the Mueller glia will only fix those cells that are lost. And we really don't know how that happens. So there's a lot to be known here, but I don't have time to talk about that tonight. So I'm going to talk about other things. My research is mostly around how do you even convince this cell that doesn't have a brain, doesn't have eyes, doesn't have ears, doesn't know anything, and can't think, to know that there's something that needs to be fixed. Okay, I would be willing to talk to anybody after the lecture tonight, but what I want to say is that to fish, their retina is a lot like our skin. Do some damage to it, it'll fix it up. Their fins are a lot like our skin, and not like our limbs. So this is a man, Mr. McKnight was in an accident while he was working on the railroad. I'm sorry that it comes out that way, but he really was working on the railroad, and suffered an industrial accident which took his right arm and both of his legs. This is usually traumatic back then. People died most of the time from this. Even today, don't try this at home. It's a good way to actually not survive. But as you can see, he did not grow back his arm and his legs. This is the kind of thing that you think, we don't regenerate from that. So something's different about fish and us that we'd like to figure out. What is it about amputees that they can't regenerate their tissue? When I was a little kid, I used to read the Spider-Man comics. And in it, there's a villain. His name is the Lizard. And he was a professor. Right? the kind of professor I want to be except for what went wrong. He was trying to figure out how to regenerate his arm, and instead, when he gave himself all of the magical cocktail, he turned himself into a lizard. And he'd go around terrorizing the world until Spider-Man would stop him. Well, they didn't make that up. There's actually an animal called a salamander that if you take off its front arm, it will grow it back. So I want to show you a time lapse of that. Here is a salamander, if I get the movie to work. Nope, gonna have to do it twice again. Nope. Go. And you're gonna have to listen to me over the woman. This is gonna take 90 days, but these are just time-lapse images of that salamander as it regenerates its limb. I want you to understand what was going on here. This is a cartoon salamander, and you can see it's wiggling around. It's got bone, it's got muscle, it's got nerves, it's got the ability to feel. So let's anesthetize it before we do something horrible to it. We're going to grab some scissors, we're going to cut its limb off.
That's nice.
Don't worry, don't worry.
It's better than yanking.
It will heal over, and then it'll regenerate. Okay, so if we could pull back the skin so we can see what's going on inside, you're going to see a bunch of cells stream over to the site where the wound was. And then they're going to push out and create a new group of cells right here. This we call a blastema. And why they've colored these green and white and red is to show you that the cells that come in here, these stem cells, come from different tissues. Some of them come from the bone or cartilage, others come from the muscle, and still more come from the skin. But none of them come from one source. We don't make the whole limb by just bringing in a stem cell and then regenerate the limb. We need 3 different types of stem cells to be able to regenerate a salamander limb. So these are adult stem cells for salamanders, and we don't have anything like that. Something about our physiology means that we control our growth to the point where we won't recover from injuries. But I would think that would be great. So I'm in this business to see whether or not we can't figure out a way to fool our cells and make them think they're salamanders and regrow us some limbs. All right, to understand what those cells are doing, though, I've got to explain what I mean by a stem cell. And I'm guessing that what I think of as stem cells is just slightly different than what you're conceptualizing right now. You're probably thinking that a stem cell has the ability to divide and make a new cell. And that's true. And you're also thinking that those cells can divide, and you've got 2, and then those 4, and then those will divide and you'll have 8. We call those progeny cells because they're the progeny of the original cell. But these cells will oftentimes differentiate into muscle or cartilage, which lays down bone, or skin. And so they're a particular kind of cell type. What a stem cell does is before it even does this first division, it does another division. And from that division comes the one that makes the progeny cells, but it also replaces itself. We call this self-renewal. And this happens for a lot of our stem cells at a very low rate. So low and so infrequently that it's hard to see them in the time frame that it takes a graduate student to do a study. So it's not particularly well studied. It just takes a long time. That's not the only way to think of stem cells, though. Some stem cells can divide, and then they'll turn into a particular kind of cell. In this example, it's a neuron. And that's the only thing that they can make. So we call them unipotent. Uni meaning one, and potent that they have potential to become just one type of cell. There are other cells that are just a little bit more useful. They can turn into multiple things. In this case, we have a white blood cell here, or we have muscle. And they all came from the same stem cell. Because that stem cell can make multiple different things, we call it a multipotent stem cell. Now forgive the jargon here. There's no difference really in English between multi and pluri, but in science there is. Due to the way that we study these things, we've also found that there's another kind of stem cell that not only gives rise to the unipotent, but it also gives rise to the multipotent. And if you can think of the cell that you would like, it can give rise to all of them. We call this cell that can give rise to all of our body cells pluripotent. And it'll be important later in the lecture on how I describe some things, just to remember that one. If you don't really remember any of the other ones, just remember pluripotent. This is a cell that can make itself, but it can make anything else too. It's got an extreme amount of flexibility. The other cells And they can do some things, but they can't do everything. And so we don't usually pay as dear attention to them. I'll tell you about some exceptions to that. Think about the pluripotent stem cell as your Leatherman tool. It's got a screwdriver, it's got pliers, it's got wire cutters. It can do a whole lot of things. And the unipotent cell is your screwdriver. Okay, I know some of you can use a screwdriver for more than just driving screws, but bear with me. What I want to say is that these unipotent and multipotent stem cells are adult stem cells. They're the kind that persist throughout our life. The thing that doesn't persist throughout our life is this very capable stem cell. To really get all of these kinds of functions, we need to go back to embryology when we had a lot more potential to become all sorts of different cells. All right. You may not have ever thought about this, but when your mother's egg met your father's sperm, that cell had an extreme potential. It gave rise to every cell that is you right now. Every single one. And even more. It gave rise to some other cells that just helped keep you in mom until you were born. I'll talk a little bit more about that in a second. So that is the totipotent cell. It's even more potent than the pluripotent cell that I told you about. And what's cool about that cell is that right after it was fertilized, it divided into two different cells. And those two cells could have separated. And if they had, your name might be Thomas because you'd be a twin. You would be an identical twin. And that's true even through the 4-cell and the 8-cell stage. If at any time these cells become separated from the rest of the group, they can go on and form a whole new person. After about 8 cells, they really start to like each other, and so they start sticking in strange ways that makes it hard to see where one cell begins and the other cell ends under the microscope. Then a little while later, about a day later, the cells of this mass start secreting some water inside. They start squishing out water between them to fill up a cavity in here. And at this point, we call the embryo a blastocyst. What's important about this stage is that although these cells could give rise to all sorts of different parts, this one no longer can. And the cells that make up this part of the embryo are now limited in what they can give rise to. So it doesn't take long before when we're existing, before we can't make everything anymore. All right, so let's talk a little bit about the blastocyst. This is, I'm sorry, I'm showing you pictures of mice here, but if you were to look at a human, the cell doesn't really look any different. So I'm using them and figuring them out. But this is a mouse cell, or mouse embryo, and it's got a bunch of cells out here in green. Outside of the green, they secrete some sugar to keep them protected from the rest of the environment. Something we call the zona pellucida. But that's not cells. That's just me marking it up with a purple marker for you to keep reference. The cells here are in green. And inside these cells, there's a space with just fluid. And so I colored it blue. There's no cells there. It's just stuff. Over here on the left side, though, there's a clump of cells. So a blastocyst is made up of two types of cells. Ones we call trophoblasts, and they're going to go and become differentiated into parts of the placenta. So this is what the fetus is going to swim around in until it finally get loose into the real world. But the embryo comes from these red cells. So these are stem cells that give rise to every part of your body. Your neurons, your heart, everything. Just not the placenta. And so if we take these cells and we grow them in a dish, separate from one another, they can't form a fetus. They can't form a baby. And this is what makes this really important. Because these cells here go on to give rise to all the cells of your body, we call them embryonic stem cells. And in the news, you're hearing them called ES cells because it takes too long to say embryonic stem. And we're lazy, and we're like the military. Everything's going to get in an acronym sometime. This is a really important part of development because if we separate the cells apart from one another and we put them in a dish together, they won't come back together and form the blastocyst. And so if we're ever to culture embryonic stem cells, we destroy the embryo. And this is offensive to people that feel like personhood develops at conception. And that makes this whole process quite controversial and was the whole reason why George W. Bush had problems when he only designated certain embryonic stem cell lines as things we could research. Right, the question here, though, is what can they give rise to? What is their potential? And the embryo comes from the red cells. Let me be clear, though. I sometimes describe this is Humpty Dumpty. If you break it, all the king's horses, all the king's men, they're not putting this back together again. So that's why we say that to get ES cells, we're destroying the embryo. Okay, so some people would argue that the personhood doesn't start at that point in development. I told you that if you separate the first two cells of development into different cells, that they'll grow up, they'll both form a blastocyst, and then those both can be pregnancy and finally bring babies to term, and you've got your identical twins that way. This isn't the common way that it's done. The more common way that it's done is that the cells develop quite normally until the blastocyst stage, and then those embryoblast cells separate into two groups. And once those cells go on to form fetuses, then they will be born as separate individuals. So a person isn't defined at conception because the cells can split and then you'll have two persons. I'm looking at you, I'm sorry. All right, people have known this for a while. Back before I was born, there was a procedure called in vitro fertilization that was developed. It was mostly developed because people who were susceptible to having children with Down syndrome wanted to eliminate those children from their possibilities. This is the way they would do it. They would in vitro fertilize an egg and grow it up to the 8-cell stage, whereupon they would remove one of the cells, and they would test that cell and say, do you have 3 chromosomes, 21? If you did, then they would put that embryo aside, they'd go to the next one. Oh, you have only 2 like you're supposed to? Okay, we'll then make that the embryo that creates the woman that's pregnant, and then their children did not have Down syndrome. So this is a process that's been done for quite a while. And like I said, if you took that cell out and cultured it right, it would develop its own blastocyst. It could be its own child and create its own person. So these cells all are very potent stem cells. All right. They're also rather manipulable. We haven't done this with humans yet, but I want to explain something that we can do with mice in a way that you can understand what we might be able to do with people. So mice come in different strains, which is just to say that we look at them and we can see that they have different colors of hair. Some of them are completely black, and from head to toe they make black hairs. Others, they're brown, and they make brown hairs from head to toe. And others, they can't make either brown or black color, so they're white. We call them albinos. What we can do is take the embryo that was generated from the mating of these 2 black mice. And if we look at their offspring, all the pups they generate, they'll all have black hair from head to toe. We can take and put aside an embryo for a minute. We take a mating of brown mice, and if we look at all their pups, we'll find out they're brown from head to toe. But we can dissociate their blastocyst, take out the embryoblast cells, and we can transfer them using a glass needle into the embryo of the black mice. Now, those foreign cells, they don't know they're foreign, so they let them come on in, set up shop, and they become part of the embryo. I can prove that to you in another slide when I show you the picture. What we'll do is we'll find a surrogate mother. This one is albino, so she's only got white hair. And we'll implant that blastocyst into her, so she becomes pregnant with the children of the other mice. And she'll bring them to term, and when that pup finally grows its hair out, you'll find out it looks like a zebra. Well, not exactly like a zebra because it's not black and white, but it's black and brown, and it's got stripes. This is what it looks like. So this is about a year old, so we waited a little while before we took the picture here. But you can see that it's got brown hairs in stripes and black hairs in stripes. So we know that its parent wasn't the white one. But truly, this is a very strange animal. It has 5 parents. It has the 2 parents that gave it the black cells, it has the 2 parents that gave it the brown cells, and it had its surrogate mother. You all have 5 parents. There are documented cases, though, of humans that have more than 2 parents. I could tell you about that later, it's not right now. So we call this animal a chimera, because in Greek mythology there was an animal that had the head of a lion, another head of a ram, and then a whole bunch of different amalgamated body parts. And since this animal has multiple parentages, we're using the same word for it. And it's a very common way to look to see whether or not genetic defects are caused by one cell or a different cell. We just mix different cell types together and make mice out of them. So we've been doing this for about 30 years, but it doesn't have the kind of heft that we need to help a person yet. Because in order for these stem cells to be useful for us, some of you needed to have put aside a few of your embryonic stem cells when you were born. I don't think any of you did that. Do you remember? Some of you, though, might have done it for your children. You might have put some cord blood aside in a nitrogen tank with a dewar, because you were thinking that maybe your children would have some kind of trauma later in their life, and you're planning for that. So you've banked that, and it's sitting over the side. I didn't even do that. No, not for any of you. You didn't do that?
Yes.
Yeah, because they know me. Most of us rely on the fact that people are generous, and they've signed on their driver's license, and they're willing to donate their organs if they're dead. So let's talk a little bit about that for a minute. Organ donation is not infrequent. One of the easiest things to do is transplant cornea. I'm going to look at you again. Because they take very well. And you don't need as many drugs to keep them in there. Some people have had artificial hearts. Some people have had heart transplants. Some people have artificial hearts right now and are waiting for heart transplants to come in. Because you don't just get a heart every day. Some people have livers transplanted. My cousins had 3 of them for some reason. They don't seem to take with him. And the nice thing about kidney donation is that you can give up one of yours. You don't have to die. And you can still help somebody else that needs one. You can do that somewhat with liver, but intestines and pancreas, you can do that usually with smaller children, not so much with older people. And the reason is because you've got an immune system. And the immune system's a little bit like ice. It's going around trying to figure out who's the legal immigrant and who's supposed to be there. If you transplant an organ, you've got to somehow fool the immune system into leaving it alone. So this is the way I like to tell you about it. Here's our cute little soldier here. He's got a killing sword, and he's going around looking at all the cells of the body. And as he does, he finds this green cell here. You see he's not looking very happy because he's found out. He says to himself, hey, that looks foreign. He's looking at a thing called the MHC class 1 molecule. Those of you that are interested in molecular biology, come back and I'll talk to you later. Let's just say he's got a little tag on him that reveals that the shibboleth is up. He's found out. He says, hey there, you've got a funny looking MHC. Are you really from this body? And the guy says, yeah, that's nothing. A busy day, officer, and look behind you. Or anything he can, but inside he's thinking, oh no, I'm found out. I'm not even supposed to be here. He's going to kill me. And that's because he is. And unless we drug that officer here so that he's in a complete stupor, then he'll take out the heart that was replaced. So a heart transplant won't take, and a person will absolutely reject it and die anyway. So not necessarily the smartest soldier, but he's doing his job, right? And so anybody that takes a heart transplant, people that take liver transplants are on incredibly horrible drugs just to make sure that they can survive the process. All right, so one of the things we thought was maybe what we could do is we could make new ES cells. And one of the ways we could do that is we'll take some adult cells and we'll rip out the nucleus from that. And we'll rip out the nucleus from an egg. If we find a woman that's just generous enough to give us some of her eggs, we'll rip out the nucleus from those because she wasn't going to have kids with them anyway, and we'll stick a nucleus inside that. And this isn't very efficient. We have to do this with hundreds of cells. But if we do that, then one of those nuclei will take. and that will become a blastocyst. And then the worry of the world is that if you take that and find a surrogate mother, you can do what they did with sheep. So this sheep's name is Dolly. And Dolly was the first cloned mammal. And they wanted to make sure that Dolly was actually a functional sheep. And so they mated her, and she produced a ewe, which is this other sheep right next to her. Just to say that this is possible that you don't just screw everything up. It's a functional sheep that can produce more. The idea with this was that sometimes you can't get your prize sheep to mate, and this would be a good way to perpetuate the genes. And so when this happened, a whole bunch of people started thinking, oh great, somebody's out there has Hitler's genome, and they're going to put that in, and we're going to clone Hitler. Or those of you that are Star Wars fans, they're going to clone Jango Fett, and make Boba Fett or whatever else. So this has been outlawed in most all the countries. So we don't do that. But the other alternative is to destroy this embryo and harvest the ES cells. And now you've got a clone of you. A tissue clone of you, but it has the same DNA as you. So if we go back to the same ICE officer and he's looking around the body trying to find who he's got to kick out of the country, he says, hey there, I'm just patrolling the neighbor for foreigners. And the other guy says, well, there's nobody here. Because he looks exactly like he ought to. And he says, I'm safe. He's not going to kill me. And I didn't even have to drug him. That's the key here. If you can give a transplant for somebody, or if you can give them stem cells that will regenerate their heart after a heart attack, then you don't have to give them drugs to make it take. You just leave the cells there and things get better. This is what we're dreaming about right now, and why we'd want to go through ES cells. The problem is, you gotta destroy an embryo. And that embryo, if you would have taken it and put it in a uterus of a healthy woman, would have become a person. And that causes a lot of people pause, find it unethical and not worthy of doing it. Still, we do have a number of ES cells that were created this way, and so what we're trying to do with them is figure out a bunch of things. One of those is, if you can take those cells, especially if you've made them from SENT, from somebody that had heart disease, you might be able to take those cells and turn them into heart cells, so cardiac muscle. If you do that, then you can put all sorts of different random chemicals on them, because you can grow up an infinite number of these cells. That's the cool thing. These ES cells never seem to die. You can multiply them into the billions of trillions, and we haven't figured out how far we can go with them. And so we won't run out of time before we can drop every single chemical that we can conceive of on them and see whether or not they kill the cells, and we find out what's toxic to hearts, or they help the cells, and we can cure diseases that would have otherwise been problematic. What's the best thing about this is most of the time we do this with mice. We go out and we say, does this kill a mouse? And no? Okay, then we'll finally try it in a person. And then we find it kills people, because people aren't mice. Or we leave out a drug that kills mice, but it wouldn't kill a person. And there's no better way around it right now. We won't take the risk of taking something that will harm mice cells and do us. So we can find some things about drug therapy this way. Some of the things we want to do is just figure out what does it take to make neurons? Because if we're going to replace somebody's photoreceptor cells, we want to make a bunch of new photoreceptor cells and not a bunch of Purkinje cells. They don't help. So we're going to find out things that cause cells to develop right. And we might also be able to take cells from people that have Parkinson's disease, which takes about 50 to 60 years to develop. And we never know who's going to do it beforehand, and then start looking at the genes of these cells. And then if we can have them in culture, we can age them. And as we age them, we can see how they progress to Parkinson's, and we can see if there's anything we can find out about them earlier than normal. All right, so if you take all those things together, then here's the potential uses of stem cells. Now, admittedly, some of these seem more serious than others. For example, stroke. If somebody has a stroke and part of their brain dies off, it would be nice to be able to replace those neurons and give them function again. Learning defects, say schizophrenia or autism, if we could figure out how to make the connections right with healthy cells, we might be able to help children while they're developing and get better. Others of them, maybe not as urgent. I know there's some men that would pay quite a lot of money to be able to put the hair back on their head, but they're not necessarily going to die over it or have a decreased ability to think and manage their life like they would if they had a stroke. But we're hoping that blindness and deafness might be cured by these kinds of cells. We might be able to strengthen hearts that have had heart attacks. And some of you have seen the Ice Bucket Challenge. If we could help ALS or muscular dystrophy, those would be things that would be on the top of the agenda for a lot of them. The funniest one, I think, here is missing teeth. Let me explain what I mean by that. You all went through two sets. You went through your baby set and you went through your adult set. And when you were a baby, those of you that had children, maybe your children have teething. You remember that that wasn't a very fun experience for them. You tried to give them Ambisol or something that would keep the pain down. I just think about my dad. You know, kind of ornery cuss every once in a while. And him getting his third set of teething teeth, right? Instead of his dentures. That could be funny, as long as I didn't have to live with it, right? So all of these things seem to be able to be functional, but we're only going to make it there if we can get through some of these ethical quandaries we have working with ES cells. So recently we found that if you take some cells from skin, you put them in culture, and you force genes into them, then what we can do is turn them into anything in the body. So when we look at them, they don't act exactly like embryonic stem cells. So we don't call them induced embryonic stem cells, but we call them induced pluripotent stem cells. The pluripotent is to tell you that they don't make placenta, but they do make everything that forms in the body. So we call them iPS cells, and they're giving us some new hope that we can go forward with this. There's a problem that we have to address with them, though. Do they work like ES cells? Otherwise, we might find that they have problems. So one of the questions is, do they have a morphology similar to ES cells? The cells we took look kind of like triangles, and they turn into circles. And do they express the same genes? So we can look at what genes are expressed. Genes are controlled by the state of the DNA. So we can look at something we call the methylome, which controls the DNA, and see whether or not the right kinds of genes are demethylated. And then we can also see whether or not the cells will, if we put them into blastocysts, can form new animals. Now we've done this with mice. I've already shown you a picture of that one. We're not likely to do that part in humans. But we can do this with the rest of those cells. And so there are some answers to this. The morphology seems to be the same. We seem to be able to make the same kind of cell types. But when we look at the gene expression and the way the DNA is arranged, we find differences. So they're not the same. And that worries us to some extent because these cells act a lot like cancer. And the last thing we want to do to help somebody overcome a heart attack is give them heart cancer. Well, maybe not the last thing. Because if they're going to die in 2 weeks, maybe we'll give them cancer in 10 years of the heart. Maybe it's still worth it. And so some of those trade-offs have to be given. And I know a number of people that have gone blind that have called up my old boss and said, can I get some stem cells? Because I'd rather be dead in 2 years than blind for 10. And so people are going to have to weigh how they're going about doing this. But it's sufficient to say at the end of my talk here that we don't know.
Travis, quick question. Go back one frame. When you talk about gene expression, that means that the gene is present or not present?
Exactly.
And the methylome means, what's the difference between the two, I guess? Is it that it's working or not working, or?
Well, if a gene's turned on or off, it depends on how the DNA looks. So if we look at the DNA, sometimes we can figure out whether the gene is going to be on or off, just the way it looks.
But it's still there?
It's still there.
Active or, okay, thank you.
It just changes a little bit chemically. So what I've told you tonight is that there's 3 kinds of stem cells. There's an adult type that regenerates your skin and other kinds of things. And then there's embryonic. And the embryonic is the gold standard of how to make everything in your body. But it's got the problem that we have to destroy an embryo to be able to get it. So our hope here is that these induced pluripotent stem cells can be done in the right way, that we can either create embryonic stem cells, or we might be able to create the right kind of adult stem cells. Those of you that are type 1 diabetic, oftentimes are just missing what are called pancreatic beta cells. And if we could just give you a supply of pancreatic beta cells, I think you'd prefer it over the daily injection of insulin. especially if we could get away with only giving you a monthly injection of pancreatic beta cells. So some of these kinds of treatments have the ability and the true potential to alleviate a lot of suffering for people. But they still have the drawbacks of we don't know yet how to control them and to make sure that they do what we want and they don't become cancer and they don't go off the deep end and start making things that we don't know. So those are the challenges that we still have and why we're spending a lot of time doing research in them. So I'll stop there and thank you for your attention. If you'd like to ask any more questions.
So, there's little hits that I'm not really sure what they're all about, but that claim that if you save your primary teeth, that there's stem cells that can be harvested from them.
Yes,
What do you think about that?
it's possible because the way that your tooth grows does go from the stem cells that form the core and then on out. Now, whether or not it's guaranteed, no, I'm not betting that far. But if you got a freezer and you have the ability to cryopreserve them with sugar and stuff like that, go right ahead. Some DNA lasts quite a while. One of my old bosses used to look at the Dead Sea Scrolls and isolate DNA out of those, and they're thousands of years old. So DNA is not so completely unstable, but I don't know that I really worry about that. My hope with the iPS cells is you won't really need your teeth cells, you just need to figure out how to shove math, I'm sorry, I'm going to start getting technical, math 1, into the right kind of cell, turn it into a primordial tooth cell, and then work it out that way. That's one of the reasons why I've never gone with cord blood with my own kids, is I was like, wow, just let technology catch up with them, and I won't have to pay any money anymore.
Yeah, because people come in and ask me about these all the time, and I'm just like, I don't know if it.
Yeah, I mean, if it makes you feel better, it's no worse than buying a new car. So I'd go ahead and do it. Mm-hmm.
There's a lot of orthopedic adult stem cell applications out there right now. Have you heard of any problems, like you said, like with adult cancers and not being able to turn off the adult stem cells?
Well, the nice thing about this is it brings to mind all of those things about fracking, for example, or other kinds of new technologies. One of the things the industry is really good at saying at the first place is, there's no evidence that this causes any harm. The follow-up question I would always like to ask those people, has there ever been a test done? Because you won't find any evidence if you haven't ever done a test. And for some of these things that we're talking about today, the reason we don't know that isn't because there isn't being a test done right now. It's just that we have to wait until it's finished to know. And that's going to take years. So some of us have to be a little bit adventurous here, right? That we're going to actually do something without knowing whether or not it's going to cause harm. We weren't going to get any of the oil out of shale if we weren't willing to try fracking in the first place. But then you need to follow it up and say, well, is there any contamination in groundwater? Same thing. If you're going to put stem cells into people's bodies, then you're going to have to follow it up and say, okay, what happens? And we'll hope that it's not the same thing we found with anabolic steroids. That, yeah, it bulks you up, and it's really good that way, but it has a whole bunch of other side effects that you might not want to deal with 10 years down the line. And my feeling is, though, the more that people are educated, we're going to find people that will accept all these different kinds of risks. The same as we find people going to the top of Everest without any kind of air support. Right? It's just part of the thrill of life anyway, and so why not go in? But it's important that people would know what they know and what people just can't know yet before they get involved in it.
I think you said this and I didn't quite catch it, but the I in IPS, the induced part, this is about taking a pluripotent cell and inducing it to do something else? Or to
No, this is to take a cell that's not even a stem cell.
Oh, okay.
And
So
inducing it to
you're inducing it to
become
become
stem
a pluripotent. Okay, thank you.
That's the cool thing about it, is you seem to have an unlimited supply of the useless kind. So let's see if we can't educate them into something good.
I really do that.
I hate my phone too. But not that bad.
If there weren't any ethical barriers, like how close are we to like solving things, I guess?
Man, I'd be paid well if I actually knew that answer. Somebody'd come over and say, what company should we be betting on? And I'd be able to place some bets down there. The problem, well, the nice thing about scientists is that they don't know it. We can act smart, we can say what we do know. What we're really lousy at is saying what we don't know. Although I'll admit it right now, we don't know. We don't know a lot of what we don't know. The only way we're going to figure it out is to start trying. But time frames are always hard. If you're a venture capitalist, it's going to take 5 years. If you're not a venture capitalist, I don't know how long it's going to take, because the venture capitalist will give me money if I say 5 years. And in 4 years, he'll forgot what I said, and it'll be 5 years then. Be worried about when somebody says, well, we're going to get it done next week, because even if today, for example, we cured cancer, you won't get the treatment for 10 years, because we're going to need to establish that I'm right. And it's going to take a decade before people are going to be convinced that I'm right. We're going to have to do that many tests. We're going to have to wait that long before people could be convinced. Oh no, oh you got better for a week. That's not good enough. How about 10 years? You got 10 years under your belt? Maybe I'll consider it. And so that's why it's really hard. It's also hard because if you're a researcher, you want to get your PhD in 5 years. So it's hard to recruit somebody into a study that won't be funded for only 3, to do something that's going to take 10 years to do. And until society starts thinking that it's worthwhile getting studies that take a decade and paying for the studies that take a decade, we will be blind to a lot of that stuff. So I'm hoping that we'll generate the political will for it. But it's hard for a venture capitalist company or somebody that's throwing money out and has stockholders that they have to deal with looking at quarterly results to get involved in this kind of thing, because their impatience is so extreme that things won't happen on that level.
How does the research you're doing compare to research done in overseas in Germany or other European countries?
I come from the Midwest, so we're kind of a hub out there on retinal regeneration. But there's quite a number of other labs throughout the world that look at different animals and how they can regenerate. So I didn't have time to tell you about things like flatworms that seem to be able to regenerate everything. You cut their head from their tail, and their tail will grow a new head, and their head will grow a new tail. You can cut off their left backside, and it'll grow everything that isn't their left backside back together. You can look at different animals, but the question is going to be always coming back to, all right, so they've got these kind of genes in common with us, and that seems to be the things that are necessary for regeneration. What is it that's stopping us? What is this added thing that we all of a sudden say, okay, we've got all those genes, but we're still not regenerating. So it's gonna take a worldwide effort. We just lost one of the greats who studied salamanders. A guy named Sonus out in the University of Dayton has been studying salamanders. He just died about 6 months ago. But luckily there's new people, and we've got a lot of increased tools to be able to do that. Some of you might have heard of CRISPR technology that's allowing us to not only get rid of genes, but replace them with other genes that we'd like. This is going to change how we test who's important, what gene is important, and what genes are not important for any process in development that we can think of. And let me be clear, development is from when you were conceived until you die. There's something going on all the time that's changing you. And so the study has to be throughout the whole lifespan to be able to get a full understanding of it.
Are there some animals that seem to be more potent in change than human beings, or is it the hierarchy, salamanders?
Yeah. Well, you can say that deer are a little bit better than us. They have the ability to regenerate their antlers every year. But no, mammals kind of stink at this. We do a lot of this kind of research in mice. Mice have a problem in the sense that they're basically day blind. They have what are called rods, but they don't have very many cones. So the fish actually looks more like us in the eye than a mouse does. But we can still look at it for regenerative properties. And we've found a few kinds of extracellular signals that if we time them right, we have the ability to delay a degenerative model, one that causes what we call retinitis pigmentosa in rats and mice. But we haven't gotten a good kick on it. We've been able to get the Mueller glia to proliferate and make a bunch of cells, but then they get stuck where they're at, and they don't move to the place where the photoreceptors would be. And so we haven't been able to get them to become sighted again after they were blind. That doesn't mean it's impossible. It just means it didn't turn out to be easy, which is what I was kind of hoping, that I'd just go in and find this factor and I'd say, hey, let's treat this drug, and we get an eye that didn't know it was damaged to regenerate some of the few cells that it lost. And somebody that had this slow degeneration would no longer have the degeneration and in fact would become better at seeing over time. Turned out none of the factors that we found are required for regeneration is sufficient to cause. There seems to be some kind of combination, and that combination is not only what is it, but when and how much, and that's making it hard to get our hands wrapped around.
I hate to interrupt this great line of questioning, but Dr. Bailey's going to be available right after if you have any one-on-one questions that you wanted to ask him. Let's give him another round of applause. Really quick request of everyone, in the back tables, there's one back there, at the very back, and this round table over here. I have these surveys that you can fill out if you have even just comments about the food. What types of food would you like, or how can we improve this? I'm really interested in your feedback, so if you could just fill this out, just take a couple minutes, and there's pens and papers in the back, and thank you all so much for coming.
Yeah, but I mean, we know all of them. I mean, it's 5 years ago. I don't remember. I don't remember all these.